Good morning or good afternoon. Hey guys, and welcome to a very short video on epigenetics. Epigenetics is the study of how the environment can directly affect our genomes or our DNA in ways that early biologists and evolutionists might have very much disliked. So if you guys remember, we talked briefly in evolution about a guy named Lamarck. Jean-Baptiste Lamarck. What a wonderful French name. And Lamarck's idea was that an organism's behavior could change its physical structure and thus its genetic structure. So Lamarck's idea was that, giraffes had started off with short necks, and that over time, through trying to stretch to reach those leaves on the highest trees, that they stretched their necks out more and more. And as they stretched their necks out more and more, when they had kids, their kids had stretched necks because they'd stretched their necks. And then the kids kept stretching, and it just kept getting longer and longer until giraffes had long necks. Now, of course, we think today, as under Darwinian evolution, we think that's kind of ridiculous, right? Because we know that long neck isn't due to behavior, right? It's due to genes that are passed on that help to have that help to build that long neck. And so we don't typically think of Lamarck's ideas as being very accurate. But it turns out that Lamarck's idea that behavior or environment can affect the genome in some way wasn't entirely wrong. And that's where we lead to epigenetics. So epigenetics is very easily misunderstood, and I don't want you guys to misunderstand it. So I'm just going to do a very brief video here on what it actually means. And I'm going to start off with an analogy because I think analogies are important and helpful in teaching science. I want you guys to think about this ball of yarn. Now, if you've ever done any knitting, or if you've known anybody who's ever done any knitting, when you want to make something out of a ball of yarn, if you're knitting or crocheting, you obviously have to do something to the ball of yarn. You can't just, as this picture suggests, you can't just have a ball of yarn, stick some needles into it, and voila, you have a scarf, right? There's some sort of in-between step. The first thing you have to do when you're knitting or crocheting is you have to unwind a bit of the ball. If you don't unwind the ball, well, sticking needles into it isn't ever going to make it turn into a scarf. You've got to unwind it first in order to be able to use it, right? And so if we look at this picture here, even though it's a little bit blurry, we can see this ball of yarn has been unwound, and that string has been wrapped around these needles in a certain way to produce a product. Okay, so in other words, once again, we must unwind the ball of yarn in order to use it to make a product. And you're probably like, okay, Mr. Garner, why in the world are you talking to me about balls of yarn and knitting if we're supposed to be talking about epigenetics and DNA? So I want you to think about a chromosome as being like a ball of yarn. Okay, it's wound up very tight, and it's made out of that DNA string. We can call it string here in this analogy. Okay, we can't use it when it's in this form. When it's as a chromosome, we can't use it. The chromosome is the packaged form of DNA that we would pack DNA up into if we were going to split the cell or transfer it someplace or something like that. But we can't directly use it. In order to use the DNA that's within that chromosome, we're going to have to unwind our ball of yarn. So here's our ball of yarn, our chromosome, and we can see that actually it's just super wound DNA. And the DNA is super round around these little balls, but we can see that it's still a double helix when we unwind it all the way. This double helix is what we use in order to make a product. If you remember, when we talked about the central dogma of biology, we said that we needed to start with a gene and DNA, and then go to mRNA and to proteins, what we just learned, transcription and translation. Now, in order to make that happen, we have to have an open double helix. So we unwind our ball of yarn, we get to our open double helix, and we can use it. Okay, why is this significant to epigenetics? Well, here's the thing. The way that DNA is wound is that it's wound around these little balls, these proteins called histones. And when it's wound around these histones, we can see it, this double helix going around and around. It can be wound tightly, or it can be wound loosely. And a little bit of DNA is going to be exposed if it's wound loosely. Okay? I want you to think about it this way. When DNA is loosely wound, it can be accessed. The double helix can be used. If it's wound very tight, though, and there's no loose DNA sticking off, then it can't be used. So epigenetics is basically focusing on two different types of groups. We have methyl groups and we have acetyl groups. Okay? And you don't really need to know much about the molecular makeup of those right now. It's beyond the focus of our course. But what you do need to realize is that basically all they're doing is they're either more tightly winding DNA around those balls to make it so it cannot be accessed, so no proteins can be made, or it's unwinding them from the balls so that the DNA can be accessed and proteins can be made. Okay, so basically it's just changing the access of your body to be able to open up those genes on your DNA in order to make something, to make a product. Just like you'd have to unwind the yarn in order to make a scarf. Okay, if you're tightly wound, you can't do anything. So methylation patterns that they were talking about there, and the addition of methyl groups, is basically saying that under certain environmental conditions, whether you're smoking or maybe you experience starvation or maybe you, I don't know, you have some sort of, maybe you drink a lot of alcohol or something else, some sort of environmental thing can methylate regions of your genome and change the genes that your body makes. And some of these changes might be good. They might be changes to make it so that you can better survive starvation. Some of those changes might be bad, such as we can see in some studies that smoking actually will change or methylate certain regions of your genome and make it so that your cells are more likely to develop into cancer cells. So here, when they're talking about this in the epigenetics video that you guys watched, what we're really saying, when they're looking at those twin studies, they compared 3-year-old twins, or whatever, how old they were, and older twins, and they were trying to see whether or not the methylation patterns, those areas of DNA that were super tightly wound and couldn't be used, changed between these twins. And they saw that, yes, absolutely, that was the case. So when we look at these twins, we can see the 3-year-old twins, the areas in yellow show all the areas that they share in common in their genome. They share almost the exact same methylation patterns. Over here, we see very little yellow, indicating that the older that these twins have gotten, the more different they have become, which suggests that changes in their lifestyle and unique things that they each do in their lives dictates how it changes their genome. So these changes to your genome, are they permanent? No. Will they be passed on to your children? Yes. Okay, so your direct descendants, your children, will inherit the molecules that will methylate similar regions of their DNA as yours. So the choices you make in your life right now and what you choose to eat or drink or do are going to affect your children. Will it affect their children? Well, it really depends. It depends on many different factors, what they experience, what environmental changes to their methylation patterns do, but it may also be able to affect their children. It can affect up to 3 generations that we know of. But that's still 3 generations of people that you're affecting by your decisions. So just makes you think, okay? Just thinking about epigenetics, basically just using molecules to change your genome based off of what you experience in your environment. It gives you a new perspective on what you might want to do and what you might not want to do and who you're affecting when you make decisions about your diet and your personal life. Anyway, just interesting things to think about. I hope that clarified epigenetics for you if you were confused about it. Of course, this is a very superficial explanation of it, and I encourage you, if you're interested in this subject, to definitely dive in and learn more. Hope you guys have an awesome day and enjoy this exploration.